Hello, my name is Allison Camilletti, and I am a scientist at BioAspect, where I lead our Drosophila Services division. In this webinar, I would like to introduce you to the benefits and features of Drosophila as a therapeutic drug discovery platform with a focus on neurodegenerative diseases. In this webinar, I will talk a little bit about BioAspect and who we are, discuss the details of our Drosophila drug profiling platform, and compare the features of Drosophila to other popular models. Drosophila is not a new model for disease. I will discuss its many contributions to science and disease throughout history and how you can use it in comparison to many human systems. What do we measure in Drosophila? I will discuss some reliable and robust behavioral assays that we have in place and how we use them to evaluate different neurodegenerative diseases such as Alzheimer's, Parkinson's, and ALS. I will then end with answering some common questions we get about our Drosophila system, and then give you an overview of what we can offer to you as our client. BioAspect is a preclinical contract research organization located in downtown Toronto, Canada. Our division of BioAspect services specializes in compound efficacy and toxicity screening of drug or nutraceutical compounds, using either in vitro cellular models or in vivo Drosophila or rodent models. Our in vitro cellular models are often the first step in preclinical drug screening. At BioAspect, we can perform efficacy and toxicity assays that measure metabolic activity, oxidative stress, viability, proliferation, differentiation, apoptosis, and others in response to drug compounds. For in vivo screening, we offer Drosophila models of neurodegeneration, diabetes and metabolism, stress and longevity, and also aging. In rodents, we also offer neurodegenerative models of Alzheimer's and ALS, and in addition have models for diabetes and obesity and for addiction. Drosophila is a unique screening platform that has many benefits that I will discuss with you shortly. But first, let's take a look at the benefits and limitations of traditional two-step preclinical drug screening approaches. We know that cellular models are fast, cost-effective, and are great for screening a large number of compounds. However, from cellular results, we are unable to infer how compounds will be absorbed, distributed, metabolized, excreted, and tolerated in an in vivo model. Because of this, it is often hard to interpret how well cellular results translate into in vivo models. We can address these issues using in vivo models, allowing us to better understand how drug compounds will be tolerated in systems that are more comparable to humans. Rodents are highly comparable to humans and great for this purpose, but they are a poor choice for primary screening of large sets of compounds. They can be time-consuming, especially for neurodegenerative diseases, as rodents need to be aged considerably before the study commences. They are also quite costly to maintain in comparison to other models. At BioAspect, we believe our Drosophila therapeutic platform is best positioned between cellular and rodent screens. Conducting primary screens in Drosophila addresses both of the limitations encountered when using cell and rodent models. Drosophila is an excellent model for narrowing down cellular hit collections even further, providing you with a more focused set of compounds to carry into the next phase. Testing compounds in Drosophila also ensures that they display characteristics of oral availability, metabolic stability, and low toxicity, all important considerations for rodent drug delivery. By moving forward with rodent testing for only those compounds found to be effective in Drosophila, it ensures that you are left with only the most biologically relevant compounds. This relieves you of wasted time and money testing ineffective compounds. Further, effective pre-screening in Drosophila will reduce the amount of rodents screened in the final analysis. This is consistent with the replacement principle for the humane use of animal models in scientific research. At BioAspect, we offer drug candidate efficacy profiling to help you determine what disease indication your drug or nutraceutical product would be most effective for. We can also perform drug repurposing to test the effectiveness of characterized drugs for different disease indications. We offer drug efficacy screening in transgenic and induced models of neurodegeneration, including Alzheimer's, Parkinson's, and ALS, and in models of diabetes and obesity. We can help you validate different molecular targets in your disease of interest, and we can help you create a Drosophila model specific to your research needs. So I have mentioned the name Drosophila a few times now, and I just wanted to provide you with more information on this fruit fly species. Drosophila melanogaster develops from egg to adult in 10 to 14 days and lives up to 120 days. They are small at about 3 millimeters and weigh around just shy of 1 milligram. Females have 2 X chromosomes, where males are typically XY. Females and males are diploid and have 2 sets of 4 chromosomes. They have a genome size of 143.9 megabytes and have 17,700 genes. If you look closely at the features of Drosophila at each developmental stage, it is apparent that this insect can be thought of as 4 models in one. Fertilized eggs, or embryos, are great models for developmental pattern formation, studying cell fate and organogenesis, and exploring how neurons and axons form and develop. The 3 different instar larval stages are great for studying early developmental mechanisms. Larvae also offer simple behaviors like foraging and response to light that are very easy to score. The pupal stage is a great model for studying the development of imaginal discs, which are structures composed of cells that are precursors to adult tissues. Then lastly, the adult fly is great for more complex behaviors and additional features I will discuss with you in more detail. Drosophila is extremely beneficial as a human disease model. Approximately 77% of identified human disease genes have homologs in Drosophila, with extremely high conservation in regions containing functional domains. They also have very highly similar organ systems to humans. For example, flies have similar cardiac, smooth, and skeletal muscle types compared to humans. There are also many conserved molecular signals and transcriptional networks involved in muscle development and pattern formation between flies and humans. Drosophila has a tubular heart composed of 3 parts that pump hemolymph around the body. Heart cells which line the human and fly heart are very well conserved, and so is the development regulation of heartbeat. Interestingly, the fly has been an extremely useful model in identifying the genetic basis of human congenital heart disease. Flies and humans both detoxify the body by releasing urine. Flies have kidney-like structures called amphibian tubules and also have conserved cells that filter waste from the blood. In place of lungs, flies have a tracheal system that oxygenates the fly's body. Remarkably, cells that line the airway epithelium are largely conserved between flies and humans. This, in addition to renewed interest in innate immunity, makes the fly a great model for asthma research. The fruit fly has saliva and also an organ called a crop that functions as a stomach. Nutrients are digested and absorbed in the gut, similar to mammalian intestines. The fly fat body stores carbohydrates and lipids as glycogen and triglycerides, which is very similar to the mammalian liver and adipose tissue. Carbohydrate levels are also regulated by insulin-producing cells, similar to a human pancreas. It is possible to induce obesity and diabetes in the fly through diet, and it is considered a great model for both of these diseases. Both flies and humans contain motor and sensory neurons, where sensory neurons exist in flies for vision, smell, taste, hearing, mechanical information, temperature, and noxious stimuli. In both flies and humans, the CNS is divided into a brain and ventral spine cord. Also, the genes that regulate nerve synapse formation and that are required for inducing action potentials are conserved in humans and flies. What about some other popular model organisms? Let's take a look at some of the features of other small models like the worm C. elegans or the zebrafish Durario and how Drosophila can address some of the major limitations associated with these models. C. elegans, like Drosophila, has a high number of disease homologs. C. elegans is also transparent, which is beneficial for live imaging purposes, and like Drosophila, has a very well annotated and well-researched neuronal network. C. elegans and Drosophila also allow for the use of genetic tools. In contrast to Drosophila, however, C. elegans lacks an immune system, heart, and has no centralized brain, thus can only produce very simple behaviors. Drosophila, in contrast, has an innate immune system, a tubular heart, and a centralized brain. It can perform a variety of complex behaviors I will discuss with you in later slides. The zebrafish also has a high number of disease homologs and has many human systems like Drosophila. Drosophila and zebrafish also have a faster developmental time compared to mice, and both lay transparent embryos that develop externally. Both Drosophila and zebrafish have a centralized brain and can produce very complex behaviors. Limitations in the model are that unlike Drosophila, zebrafish has limited genetic tools available for its study. Also, the cost of laboratory infrastructure and maintenance fees associated with studying zebrafish are quite expensive. In contrast, Drosophila has many genetic tools at its disposal that I will discuss in more detail with you in later sections of this webinar. Also, the laboratory infrastructure associated with Drosophila is much cheaper compared to zebrafish, and flies are much easier to maintain. Drosophila can also be easily genetically manipulated. Transgenic lines come in many varieties and can be made from human constructs, insertion-deletion mutations, or other allelic variations. This allows us to again target your gene or disease of interest. Further, because many human genes show strong conservation between flies and humans, you can test your compounds in a system that has conserved disease pathways and pathway components. One of the top benefits of Drosophila is the array of genetic tools at its disposal. We can increase or knock down gene expression in certain tissue types and at particular developmental times. We can also create populations of cells with different genotypes using mosaic analysis, and we can perform precise DNA editing using CRISPR-based systems. All of these techniques allow for a deeper level of analysis and mark targeted changes in gene expression. Using Drosophila, we can capture a range of phenotypic endpoints. We can measure different aspects of morphology using immunohistochemistry, we can measure body size and weight, and we can also measure ommatidia patterning using a pseudopupil assay. We can also track longevity and measure developmental time, and we can measure different behaviors, including learning and memory, courtship, motor function, and olfaction. At BioAspect, we can quantify different combinations of endpoints on the same or different fly cohorts. We can also measure phenotypes over time and at different life stages to capture disease progression. As I have mentioned previously, using the fly as a model of human disease is not new and has led to many important scientific discoveries throughout history. In 1901, William Ernest Cassel used flies instead of mice and guinea pigs to study coat color because they were easier to breed and much less expensive. In 1933, Thomas Hunt Morgan, considered to be the founder of Drosophila genetics, received the Nobel Prize for showing that chromosomes carry heritable genetic information, and he used a fly to do it. In 1946, Hermann Euler won the Nobel Prize for his fly research showing that radiation could produce genetic mutations, such as chromosomal rearrangements. In 1971, Seymour Benzer discovered the period gene, a gene of critical importance in fly and human mammalian circadian rhythmicity. In 1995, Nielsen, Volhard, and Lewis won a joint Nobel Prize for discovering key genes that control embryonic development. Many of these genes are considered. In 1995, Nielsen, Volhard, and Lewis won a joint Nobel Prize for discovering key genes that control embryonic development. Many of these genes are conserved in human developmental pathways. In 1998, the first model for the progressive neurodegenerative disease of spinocerebellar ataxia type 3, SCA3, or Mercado-Joseph disease, was created in the fly. This disease is caused by repeat expansion of a single gene and is characterized by locomotor dysfunction, speech difficulties, tremors, and eye problems. The Drosophila genome was sequenced in 2000. This greatly accelerated Drosophila research and made the fly an attractive model for scientific investigation. More recently, in 2011, Hoffman, Batula, and Steinman won the Nobel Prize for their research on the role of toll receptors for fly innate immunity. Toll-like receptors are also present in humans, where they play a large role in adaptive immunity. The fly has a long history of being a robust model of human diseases, and today fly models exist for a large range of diseases, including the ones that we work with, like Alzheimer's disease, Parkinson's, Huntington's, ALS, diabetes, and more. How do we make a Drosophila disease model? Well, making a Drosophila disease model generally involves two different processes. First, we could genetically manipulate homologs or orthologs involved in human disease-causing pathways. Secondly, we could use transgenics to introduce human gene variants into Drosophila tissues of interest. Once we have the models in place, we then analyze for the presence of disease phenotypes. We can do this using behavioral screening for learning and memory, courtship, motor function, or olfaction. We can look at aspects of morphology through immunohistochemistry. We can look at body length and weight, or the pseudopupil assay. Again, we can measure longevity and developmental time. We can also measure different biochemical endpoints using PCR, ELISA, and other techniques if needed. After we have a model and a phenotype of interest, we then perform rigorous validation of the model. This includes a minimum of 3 full experimental replications. We also use 3 different experimental operators to conduct a particular assay. We always use the appropriate parental and genetic background controls, and we use a high number of flies per replicate, and typically use about 6 to 10 replicates per experimental group. Let me just talk about some of the robust behavioral assays that we have in place at BioAspect. We can test locomotor function in a counter-current apparatus. Here, flies are given 5 chances to successfully climb into the next subsequent tube. At the end of the assay, which takes around 2 minutes to complete, the number and location of the flies are measured, and a climbing index is calculated. In the video shown here, you can see one of our experimenters conducting this countercurrent apparatus. This assay offers reproducible results that are more informative than standard one-pass climbing assays. We can run multiple flies at once, allowing for higher statistical power and lower variation. We can also test learning and memory using a courtship suppression assay in a courtship chamber shown here. In this assay, males are trained to refrain from initiating courtship by being exposed to a previously mated unreceptive female. After a training period, males are again exposed to the female and their willingness to mate with her is assessed. Males who remember rejection will not show conventional mating signs, while those that don't will still attempt to copulate. AD males, for example, those with Alzheimer's, tend not to remember rejection and continue attempting copulation. We use assays like these in our Drosophila models of neurodegeneration. Drosophila are great models for diseases of this kind because they have a complex behavioral repertoire. They also have a brain with more than 100,000 neurons and exhibit different behaviors like circadian rhythms, sleep, learning and memory, courtship, feeding, aggression, grooming, and flight navigation. They also have structures that we can investigate for neurodegeneration. Most notably, they have mushroom bodies that are like our hippocampus and that govern learning and memory, and a compound eye that's tissue is routinely used for the expression of neurotoxic genes to study neurodegeneration. Flies also degenerate with age, much like humans, allowing us to capture phenotypic changes as a function of age in less than 2 months. The brains of flies and humans are highly specialized and subdivided into functional centers for vision, smell, motor coordination, learning, and many other behaviors. In this next section, I will discuss how Drosophila can be used to investigate Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis. If you would like to skip ahead to a particular disease section, please click on the link provided on this screen. If you are having trouble finding the associated links, please make sure the annotation feature is enabled in the settings link at the bottom of this video. Alzheimer's disease is the most common neurodegenerative disease and currently has no cure. It affects approximately 1 in 9 Americans over the age of 65. This disease is on the rise, and it is estimated that by 2050, the incidences of the disease will triple. This disease is characterized by a progressive decline in memory and cognitive abilities, and pathologically by the formation of amyloid plaques and neurofibrillary tangles. The formation of amyloid plaques begins with the improper cleavage of the amyloid precursor protein, APP. Normally, APP is cleaved by alpha secretase, producing a protein product that is neuroprotective in its function. Dysfunctional activities of two proteinases, gamma and beta secretase, generate aggregation-prone peptides called amyloid beta that form these amyloid plaques and lead to the neurotoxicity seen in Alzheimer's disease. Flies have a homologue to the human APP gene, called APPL. The Drosophila APP gene, however, lacks a beta secretase cut site, and flies also lack the beta secretase enzyme. They do, however, have gamma secretase that comes in useful in making a transgenic Drosophila model. In flies, we often use something called a Gal4 UAS system, which is borrowed from yeast to drive tissue-specific expression. Briefly, the system involves coupling a known promoter and its corresponding transcription factor to a gene and tissue type of interest. Using this approach, we can target different genes to different tissues. We often like to use neuronal-specific expression for genes implicated in neurodegeneration. To make the fly work for us as a model of Alzheimer's disease, we can do one of two things. First, we could express a modified form of human APP along with the beta secretase enzyme that flies lack to induce the expression of amyloid beta 40 or 42. Secondly, we could directly express the genetic sequence of an amyloid beta isoform itself, giving us a similar end result. We tested this first transgenic model using our countercurrent apparatus that tested locomotor ability in these flies. Human APP and BAC1, beta secretase, was expressed in all fly neurons, and they were measured for their climbing ability on day 7, 14, and day 21. Compared to parental controls, transgenic Alzheimer's flies had significant motor defects on all days tested. Another research group tested these same transgenic flies in a courtship apparatus. First, they tested their ability to learn rejection by comparing their performance towards a previously mated female in the first 10 minutes and the last 10 minutes of an hour-long training interval. They found that all flies equally attempted to court the female in the initial stage of the training, but by the end of the training interval, they could significantly learn to reject the female. 80 flies, shown here as ELAV, which is the neuronal driver, driving human APP and BAC1, appear to have no problem learning rejection. In phase 2 of the study, researchers tested the fly's memory ability. Here flies were sham trained without a female, serving as a control, or trained with the rejected female. Now presented with a new female that was receptive to mating, sham-trained flies courted more often compared to trained flies, which was expected. In contrast, Alzheimer's flies showed no difference between treated and controls, and the trained flies courted females much like controls, suggesting that they did not remember the trained rejection. Interestingly, treatment with an appropriate drug can rescue this memory effect. Now let's explore another hallmark of Alzheimer's disease, neurofibrillary tangles. These tangles are formed by the accumulation of hyperphosphorylated tau protein. It is thought that the formation of amyloid plaques preempts the formation of neurotoxic tau, and then may further enhance amyloid beta toxicity. Expressing human tau in Drosophila causes a host of relevant phenotypes. Neuronal overexpression of wild-type or mutant human tau is neurotoxic in flies, evidenced by vacuolization in the brain, tau accumulation, yet no NFT, and reduced lifespan. The overexpression of human tau in motor neurons causes disrupted axonal transport and locomotor defects. Co-expression of human tau with the fly homologue of GSK3 beta enhances neurodegeneration and induces the formation of interneuronal NFT. Coupling human amyloid beta 42 and human tau induces neuronal dysfunction and decreases longevity. Here are some results that show the differences in eye morphology when human and the fly homologue of GSK3 beta shaggy is expressed specifically in eye tissue. In the top panel, we can compare control eye morphology in flies expressing an eye-specific driver called GMR with flies that have eye-specific expression of human tau or human tau plus shaggy. In both of these single or double transgenic fly lines, gross eye morphology is visibly affected. This same trend is evident when we look up close at the patterns of photoreceptor cells. We see that increased expression of tau and tau plus shaggy causes disrupted pattern formation indicative of neurotoxicity. If we look up close, we can see that the pattern of the actual photoreceptors changes as well. This is evident in the bottom panel. In the control, we see this nice pattern of ommatidia in the fly eye. However, this changes significantly, again, once we express wild-type tau within the eye tissue, and gets even worse if we express wild-type tau along with JSK homolog. This is a great way to measure the neurotoxicity of neurotoxic genes in unessential tissue. Aside from APP and tau, there are many genes that have been implicated in human Alzheimer's disease that share homologs in Drosophila. This allows for customizable Alzheimer's models for your pathway of interest. Now I will be discussing Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease after Alzheimer's and affects approximately 6.3 million people worldwide. The most common behavioral symptoms associated with the disease are slowed movements, muscle rigidity, resting tremor, and postural instability. The most common pathological symptoms associated with the disease include dramatic loss of dopaminergic neurons, a decrease of dopamine content in the striatum, and cytoplasmic inclusions of aggregated proteins called Lewy bodies. What are Lewy bodies? Lewy bodies are mainly composed of alpha-synuclein and ubiquitin. Many instances of familial PD have been linked to mutations in the alpha-synuclein gene region, leading to an increase or misexpression of this protein. Expression of human wild-type and mutant forms of alpha-synuclein in Drosophila recapitulates many PD phenotypes, like reduced motor function, loss of dopaminergic neurons, and the formation of Lewy body aggregation. We can actually look at this reduction of dopamine in expression in fly brains. Dopaminergic neurons have stereotypical patterns in the fly's CNS that we can use to measure deviations or changes. Here we see that in control brains, dopaminergic neurons are present in these distinct clusters, indicated with the arrow. In brains of flies that have increased expression of alpha-synuclein in all neurons, they have a dramatic loss of these dopaminergic neuronal clusters, evidenced by the absence of these clusters in the PD fly. Aside from alpha-synuclein, two other important genes, Parkin and Pink, have been implicated in Parkinson's disease. Normally, Parkin and Pink serve to protect cells from mitochondrial dysfunction, initiating mitochondrial death. In the Drosophila homolog of Parkin, Park2 is knocked out in Drosophila and causes loss of flight muscles and increased sensitivity to oxidative stress. Drosophila mutant for Pink1 show muscle degeneration and mitochondrial phenotypes similar to those seen in humans. Another way to model PD in Drosophila is to expose it to environmental toxins known to cause Parkinson's disease in humans. Certain pesticides and heavy metals have been associated with an increased risk of PD in humans. Paraquat in particular has been shown to cause PD. If you feed this toxin to flies, it actually induces Parkinsonian-like symptoms. Treatment of flies with paraquat leads to shortened lifespan and motor defects. This effect can be rescued through treatment with L-DOPA. This model is great for understanding how environmental toxins cause neurodegenerative disorders. Let's take a look at some results with Parkinson's disease. Here again, we used our countercurrent apparatus to show that flies exposed to paraquat have a severe decrease in motor function that progresses with age. This is in comparison to flies that have not been treated with paraquat. ALS, or Lou Gehrig's disease, is the third disease I will focus on in this webinar. ALS is characterized by motor neuron death, resulting in a decline in muscle function. This ultimately leads to paralysis, speech deficits, and respiratory failure. Pathological features include cellular aggregates, mitochondrial abnormalities, axonal abnormalities, dysfunctional glutamate signaling, and oxidative damage. There are many candidate genes for ALS that can be examined in Drosophila. One of the most popular genes is for superoxide dismutase, or SOD1. When wild-type or mutated versions of this gene are expressed in fly motor neurons, flies show progressive climbing defects and protein aggregation in motor neurons. Expression of vesicle-associated membrane protein, associated protein B, or VAP-B, in all neurons causes aggregate formation, visible neurodegeneration, locomotor defects, and a decreased lifespan. Let's look at some results from these different ALS models. Here, Watson et al. Measured the severity of aggregate formation in thoracic ganglia for wild-type human SOD and 2 different human SOD mutant lines. He found that the mutant line for G85R in particular showed severe aggregate formation in a high percentage of thoracic ganglia. A second group of researchers looked at the expression of VAP-B in larval muscle tissue using a G14 Gal4 driver. This appeared to induce visible aggregate formation of VAP-B, which is visible by immunostaining with the VAP-B antibody. In the second panel, these researchers show that these VAP-B aggregates are also ubiquitinated, suggesting they are targeted for degradation. Other relevant models of ALS include Drosophila transgenic for TDP43. When this gene is expressed in all neurons, it causes reduced adult survivorship and adult and larval locomotor defects. This gene also causes neuronal changes at the larval neuromuscular junction, driving the expression of wild-type and mutant forms of HFUS, cause reductions in adult lifespan, locomotion, cytoplasmic aggregate formation, and neuromuscular junction phenotypes. Let's look at the larval neuromuscular junction to see the effects of expressing TDP43 in all neurons. Here we can see these long branches and high Bouton numbers in control larvae. However, in the transgenic larvae, we see this reduction in branching and an apparent decrease in the number of Boutons. This is indicative of neurodegeneration at the larval stage. Now I'd like to address some common questions we get at BioAspect about our Drosophila services. How are drugs delivered? Different drug or nutraceutical compounds can be delivered through injection, either through intra-abdominal or intrathoracic injection. Through feeding, we mix our compounds into the food directly or put it on filter paper to be consumed by the flies. We can also vaporize different compounds and expose the flies to the vapor through nebulization. How is dosage calculated? Compound dosage is calculated based on average fly body weight. This is dependent on the sex and strain of the fly. We also factor in that the average adult food consumption is approximately 2 microliters per day. We often suggest testing a minimum of 3 dose levels per compound. How do Drosophila models compare to rodent models? This is highly dependent on the disease of interest. In some cases, Drosophila can actually be more useful than rodent models. For example, Drosophila alpha-synuclein models show dopaminergic neuron loss and the formation of Lewy body inclusions. This is in comparison to transgenic rodent models that do not show dopaminergic neuron loss and only show a variable presence of Lewy bodies. It is important to remember that both Drosophila and rodent models largely rely on transgenics and gene misexpression to generate disease phenotypes that actually aren't naturally occurring in either model. If your study plan involves proceeding to rodents following Drosophila screening, we can help you select the most comparable models. How long do Drosophila experiments take? We require about 1 month lead time for in-house fly stocks, and slightly longer for those that need to be ordered or customized. On average, it takes about 1 month to run a particular assay. How much do these experiments cost? We charge around $5,000 Canadian per assay, using one compound at 3 different dose levels. The behavioral assays we can perform include locomotor activity, we can test learning and memory, feeding behavior, phototaxis, and also analyze longevity. We can also generate additional biochemical endpoints per client request. At BioAspect, we offer drug candidate efficacy profiling. We can characterize the biological activity of your pharmaceutical or nutraceutical agent in different disease indications. We can support drug repositioning in new therapeutic areas and also observe off-target effects and evaluate safety. We can also perform efficacy screening in transgenic or induced models. We can assess the therapeutic effect of pharmaceutical or nutraceutical agents at single or multiple dose levels in Drosophila neurodegenerative, which includes Alzheimer's, Parkinson's, and ALS, or diabetes and obesity models, using various survival, behavioral, and biochemical endpoints. This is ideal as a secondary screen for lead optimization and can be used in target-based and phenotypic screening. We can also perform target validation. We can validate molecular targets in your disease of interest utilizing a wealth of Drosophila-based genetic techniques and biochemical analysis. We can also help you create, validate, and customize Drosophila models of different diseases. We can help you create your own Drosophila model featuring human disease phenotypes of interest for your drug discovery project. If you have any questions, comments, or inquiries about what you heard in this webinar, feel free to contact me either by phone or email. Thank you for listening.